Today, I'm going to talk you guys about how crowdsourcing single-cell data allow us to better understand biology. And as Dr. Liu presented before, I am associated with the Livestrong Cancer Institute in the Department of Bioinformatics at the University of Texas at Austin. And I'm going to start with a little background about the advantages of single-cell high-throughput technologies. So single-cell allow us to group cells by the different cell types that are present in the tissues. And when we cluster them properly, they allow us to identify the associations between different genes. So as you can see in the panel A of the figure, when cells are not grouped, we cannot detect the associations between the gene A and gene B, but when they are properly clustered, the associations are very well recovered. Single cell also allow us to identify the different changes in the regulation when the cells are undergoing certain pressures. So if you perturb your cells, they may proliferate differently, they may change their profiles of gene expression, and they may have different patterns in the composition of the tissue. So when you have tissues in early development, the composition of cells may be different than the one that is in the already measured tissue. So you can quantify the differences in the composition, but you can also order the cells based on the developmental processes. And you can trace that using a real time where you do the experiments at different time points, or you can actually compute a metric called the pseudo time to organize the cells within a certain time. So that is really, really nice because we now have the highest resolution of the processes happening in the tissues. But this resolution came with a cost. And the cost is that these methods are really sensitive. They are very sensible to be biased to the different processes of tissue preparation, the sample management, the sample sequencing. Every change that you do in the procedure may at the end affect the profiles of expression from the cells. So that is a problem. The problem is that if we have biased profiles, we are uncertain if we have recovered the patterns that are being happening in the people tissues, or they are just a mistake, an experimental mistake that happens in the wet lab. So for example, in this paper, we tried to identify the most similar cell line that is present in the patient's tumor. And if we have biased profiles in any of the cases, biased profiles in the cell line or biased profiles in the sample's tissue, we will not be able to accurately identify which cell line is present in the tumor, and then the prioritizations of the drugs to treat the patients will be wrong. So how can I be sure that my profiles are not an experimental mistake? We can keep a very close eye on the quality control. One of my experiments is biased or not. But what happens when all my experiments could be biased? Or how do we find a limit when we know what metric to use to identify the high quality cells? So for example, in single cell RNA-seq, we know that the concentration of mitochondria are highly related with the apoptosis stage of the cells. But how to find the right limit, or how to collect enough samples when they are very rare in the tissue? For example, if I am interested in characterizing a specific cell type, but this cell type is present in very low concentrations in the tissue that I'm interested, how can I increase that sample size to better characterize it? So all these problems may be solved by using crowdsourcing data. And crowdsourcing data is not other thing than just pulling up all the experiments done from different scientists and deposit in a database where they are homogeneously processed, and that allow us to identify patterns that are conserved across tissues and across studies and across species. So if we do crowdsourcing of data, we can actually find parameters for quality control, and we can increase the sample size to better characterize the cell types that I'm interested. So the source of the data for this case, what I'm going to present you today, is the PanGlaudDB database. The PanGlaudDB database was presented in 2019 from Oscar Frasen in the Karolinska Institute, and it provides single-cell RNA-seq data from more than 1,000 experiments for mice and 300 for humans. And this database is really nice because everything was processed from the FASTQ files in the SRA database. So the database also includes a search panel where you can search for a specific gene, a specific cell type, or combinations using Boolean rules. So you can decide to have gene A and gene B, or gene A and not gene B, and it will return you all the clusters where the cells express that set of genes in that particular rule. So the problem is that each one of the data sets is reported in one web page. So if I want to access to this specific sample, I will have to click here in view, and it will give me something like this. So this web page will allow me to download the count matrix, the identify clusters, the cell types, and all the genes that are expressed in each one of the clusters. But that is a manual work. I will click here to get the counts. I will click here to get the clusters and the cell types. And then I will be able to analyze the data in my own computer. But what if I want to do a meta-analysis with 1,000 of experiments? I have to do that manually. The database don't provide an API that make it easy to download all the data in just one click. And everything needs to be done through the portal. So that is what motivated the development of rPanGlaudDB. rPanGlaudDB is an R package to download and merge the labeled single-cell RNA-seq data from the PanGlaudDB database. It was done in a collaboration with my PhD supervisor, Dr. James Kai, and Dr. Kujer, who was my first postdoctoral advisor. And this tool allows us to filter cells by organism, by cell type, by Boolean rules, just as the database, download all the data, and return a surrogate object that allow the post-processing of the data in your own computer. So I will show you 3 applications of the rPanGlaudDB in a crowdsourcing fashion, and how it helped us to improve our analysis and to get biologically meaningful results. So the first application is on the mitochondrial content. So by the 2020, at the beginning of the pandemic, there was a question on how to actually filter out low quality cells based on the mitochondrial ratio. And there was not consensus on what filter to use. Most of the people say that they don't filter the data, others say that they use 5 or 10%, and others go up to the 15%, but there was not a consensus. So what we did was that we used rPanGlaudDB to systematically analyze more than 5 million cells in 1,300 experiments. We download each one of them, and we download also the labels. And what we found is that the tissues from mice have lower levels of mitochondrial content than the ones in the humans. As you can see in this picture, the difference is significant. And then the next question is like, is this an artifact, or it is actually happening as in the biology? So we divide this pattern that we see in here into different technologies to sequence the data. And as you can see, in the cases where both organisms are sequenced using the same one, like in Drop6, CU1, Fluidim, and Tenet's commune, in all the cases, the mice samples have lower content of mitochondria as compared with the humans. So what we found is that the 5% that was established as a default in several of the tools to analyze single cell data is appropriate for mice tissues, but not for human ones. So then, to validate our results, we separate this line of data from the Chromium system, because most of the cells came from this technology, to identify which tissues are below the threshold or which ones are above the threshold, and to validate if this threshold was accurate. The same for humans. So in mice, we analyzed 141 different tissues. And what we found, the number of cells for each tissue is placed in the parentheses in here. So what we found is that almost all the tissues, with exception of 4 of them, are above the 5% threshold. So it works very well in mice. It is a very good threshold to identify low quality cells. And in humans, a lot of the tissues are above that 5%. So it is not suitable to identify low quality cells from the mitochondrial ratio. So what we propose is that we could move that threshold to a more higher one, the 10%, and it seems to be working better than the previously used to identify high quality of low quality cells. Then we conclude that the differences in the mitochondrial content between humans and mice are different, that this is not an artifact of the data, and in all the cases where we have the same organism, the two organisms sequenced in the same platform, the pattern is consistent, that the 5% works better for the mice, the 10% works better for the human, and that omitting the quality control leads to wrong results in the biological interpretations. So my second application of the ARPANGLAO-DB tool is in the fibrocytes fashion. So fibrocytes are blood cells, specifically blood macrophages, that when the tissue is hurt, they transdifferentiate into a different phenotype that produce collagen. And this helps in the healing of the tissues, but it also can go out of control and produce diseases related with fibrosis. So when I was a PhD student at Texas A&M, there were 2 professors in the biology department that identified the amyloid serum P protein as a determinant to reduce the induction of macrophages to fibrocytes. And when COVID started, they tried to apply this technique to reduce the fibrotic phenotype in the patients very affected with the disease. So I was in a talk where Dr. Gomer was presenting his work, and I was very interested because there was no transcriptomic profile of fibrocytes available at the time. And this was because only 8%, 0.8% of all the macrophages transdifferentiate into that phenotype. So fibrocytes, as I explained before, fibrocytes are hematopoietic cells that acquire another phenotype that is more likely to be a fibroblast, and they produce collagen for that. So the markers to identify them are a combination of fibroblast markers and also macrophages markers, and it is a Boolean rule. So I have all these genes, but not this one. So I applied that rule in the PanGLAO-DB database, and I downloaded all the fibroblast cells that were wrong label because the database don't have fibrocytes as a specified cell type. And I downloaded all the fibroblasts that express this Boolean rule of genes. And what we found is that there was a cluster, the cluster 8, that after pulling the 2 data sets with fibroblasts exhibiting that specific set of genes, are enriched with them. So the cluster 8 in comparison with all the other clusters of fibroblasts from skin, are exhibiting the genes I'm interested in. Then to validate that they are actually clustering together, we plotted using the nebulosa package. The nebulosa package do kernel-based identification of cells. So it identifies where the cells exhibit the genes you are interested in high levels. So as you can see, this is the cluster of cells that exhibit all these genes that are expected to be characteristic of fibrocytes. So we did that, and we also did differential expression analysis of the cluster 8 against all the other clusters in the samples. And what we found is that a set of upregulated and downregulated genes are present, and the ones that are upregulated are associated with different biological processes that are highly associated with the wound healing. So the regulation of the extracellular matrix, the epithelium to mesenchymal transition interaction, the extracellular matrix interaction, sorry, the prostaglandin biosynthesis and regulation, the notch signaling pathway, the interleukin-5 regulation of apoptosis, the oncostatin M, and of course, because they are hematopoietic cells, compared with the fibroblasts, they are enriched with genes associated with the hematopoietic cell lineage. So they are involved in the inflammatory response pathway, and this confirms that association. But when we try to validate these associations of the upregulated genes, we use two approaches. We use traditional gene cell enrichment analysis, comparing the fold changes and the p-values associated with it. And we found that they passed the 5% FDR threshold. And when we did single sample gene cell enrichment analysis for each one of the cells, and we plot the same pathways, we found that only the prostaglandin biosynthesis and regulations are associated with the specific function of the fibrocytes. So far in this project, what we did was that we collected a set of 157 fibrocytes using a Boolean rule and collapsing all the data from the database. And we report the fierce unbiased transcriptome profile of the fibrocytes, and we confirm that this cell type is associated with the healing of the cells, of the tissues, when they are damaged or infected through the activation of the prostaglandin biosynthesis and regulation. So the third application that I'm going to show you is associated with the functions of the genes. So when I was a student, the Boehringer Ingelheim had this platform called OpenMe. And OpenMe is a platform when the biopharmaceutical company open a question and the people propose solutions to it. So in this case, they were interested in how to decipher the molecular targets of the human myeloid, the growth factor, so the MyDGF gene. So when I graduated with my PhD, I developed a family of tools to compute and generate gene regulatory networks using machine learning algorithms. So these tools allow us to take the data from the count matrices of single cell from a specific cell type and construct a gene regulatory network based on the associations between the genes. And this, because the single cell data is highly heterogeneous, what we do is we construct a set of different networks, subsampling the input. And then we put all these networks into a tensor to identify what is consistent between them. And this consistency allows us to produce a high quality gene regulatory network that is the noise for the different states of the cells in the population. And because this is highly accurate and it is the end of analyzing all the data together in a tensor way, we can actually identify what will be the perturbation or what will be the most affected genes when a perturbation happens in this gene regulatory network. So this is SC10-fold-KNK. It's a tool to perform in silico knockouts and predict the function of the genes through the perturbation of gene regulatory networks. Then we perturb the gene regulatory network, we aligned as a geometric, as two geometric surfaces, and then we measure the distance of each gene in the network that when they are aligned. So if the distance should be associated with the specific results. So when you align two networks, if you consider them a surface, each gene will be matching with other gene in the perturbed network. And if the gene is gone, it will cause a difference in the alignment. And this difference in the alignment is what we measure to identify which genes are differentially regulated. So we apply this to several examples, and I'm going to present you one where the authors of the data set knockout TREM2. So they identified that the TREM2 knockout in microglia is associated with the Alzheimer's disease, the oxidative phosphorylation, the metabolic cholesterol, the cholesterol metabolism, and the lysosome. So we took the wild type data from the microglia that they produce to do the comparison and identify the affected pathways. We construct a gene regulatory network and we perturb the gene regulatory network removing TREM2. When we do the comparison, we found that at the top of the list, we have the Alzheimer's disease, the oxidative phosphorylation, the lysosome, and the cholesterol metabolism. When we identify these genes and we use the hypergeometric test to identify their functions, you can see here the FTRs associated and in parentheses the number of genes. This plot, what I call a peacock plot, is colored based on the function identified in here. So microglia is known to be associated with immunity, and all the other phenotypes that or specific biological processes are in here were recovered by the original authors. We cross-validated these findings using gene cell enrichment analysis, and as you can see here, the enrichments are significant and located at the top of the list. So what if the Boehringer Ingelheim company is interested in to know which one is the function of this gene? Why not use a crowdsourcing approach to download all the cell types where this gene is expressed and identify what is the specific function that is consistent across all of them? So the first thing we did was to use the endothelial cells. That is the model that has been used before to identify the function of this gene. And we validated the specific functions identified in this cell type. So they report, the authors of the paper reported that my DGF is associated with the cell cycle, the apoptosis, the VEGF-A transcription factors pathway, and the MAP kinases signaling pathways. So once we confirmed that they actually work in the endothelial cells, we moved forward and downloaded all the cell types where the gene is expressed. We constructed the networks and perturbed the gene regulatory network, removing my DGF. We produce a perturbation profile for each one of them, and we compare the similarity of them using the Spearman correlation coefficient. This heat map present here shows you the association between all of them. So you can see that pretty much all the perturbation profiles are the same, except these two specific cell types where the gene is expressed, but the function is not consistent. So when we pull out the data and we construct a heat map, we identify that the most consistent pathways are the AKT signaling pathway, the endothelial nitric oxygen activation, the nitric oxide metabolism, the apoptosis pathway, the muscle construction, and the ALQ2 pathway. So by crowdsourcing data, we were able to identify pathways that are consistently associated with the knockout of this gene. So in conclusion, we have that SC24KNK can recapitulate the results from a cell type specific gene function, and that the pathways that I identify are consistent functions of my DGF in different cell types. So this is what I have prepared for today. And my conclusion in the final is that the crowdsourcing of the single cell data and using and developing new software to analyze it help us to understand better biology using automatic cross validation that came up from the use of different data sets and different procedures to generate the same profile of data. So to end, I would like to acknowledge the work of my PhD supervisor, Dr. James Kay in Texas A&M University, Dr. Marieke Cujer in the University of Oslo, who made possible the development of ARPANGLAUDB, the members of the KaiLab that helped to the development of the SC24 family of tools, the University of Texas A&M, the Norwegian Center of Molecular Medicine, and the Livestrøm Cancer Institute, where I work right now. I will now be open to have your questions, and here is my contact information.
Great, thank you. Very interesting talk. So please raise your hand if you have any question, or put your question in the Q&A. I think we have one question now. Okay,
yes.
We have one question from Ashish. How do you take care of the batch effect in your analysis?
Yeah, when we use crowdsourcing, we need to account for that. So I am an R developer. I pretty much use Harmony all the time. So we use the IDs of the samples where the data come from to reduce the batch effect and integrate all the samples. But this is just for visualization purposes and for normalization ones. And after that, we use the already defined and corrected for batch effect profiles to perform the differential expression and the different analysis that we do afterwards. So Harmony will be the answer for that question. We use Harmony.
Right, we also use Harmony to do the batch effect for several projects also. So maybe this is a follow-up question. Have you tried to use the, I think Surat integration function can also, remove the batch effect. Have you compared this kind of function with the Harmony?
I did use the weighted near neighbors approach. That is the one that is integrated in Surat, and it was more expensive computationally. So if you have thousands of data sets, it will be really hard to integrate. But the day before yesterday, they released a simplified version or more computationally efficient that can do quickly. So I'm looking forward to test it and see if it improves. But yeah, I cannot use that before because of the size of the data sets, but now we can do that because there is a new version that is more efficient.
Great. We have another question. Okay, we have, maybe let me ask one more question first. I think we have two more questions. So the question is that I'm really actually glad to hear that you actually do some study to give us some idea what cutoff we should use for the mitochondrial genes, for the single cell RNA-seq analysis. So 5% for the mouse data and 10% for the human data. I was just wondering, do you also find any patterns, say for example in mouse or in human data, some like cancers, for example, cancer sample, we might have, the cells maybe is more transcriptionally active. So should we give like a higher, cutoff for the mitochondrial
Yeah,
genes later?
the people have been always reporting that the new version of the tenet system, for example, the version 3, produce more mitochondrial reads than the version 2. And some of them associate it with that the protocol has a stop, like a step where the cells are in the cool bath in for like 10 minutes. And this changed the protocol compared with the version 2. So I didn't find a pattern in the cancer cells that are highly associated with the mitochondrial content, but I guess it's because the database that I used to analyze the data is not a cancer-based pathway. So it will be really nice to expand the analysis to a database focused on cancer and see if there are cancers that have more mitochondrial content in it.
Got it. I think Ashish actually have a follow-up question. I think even though you answered his question, he basically asked like, have you checked the thresholds for the data cells and double it?
Yes, like we did a linear regression analysis to identify the 95 confidence interval of the prediction. So we remove everything what was above the average of all the cells in the samples and the ones that were below. That was done before start identifying the patterns of the mitochondrial content, and that is why the threshold in here is set up as a boundary of the 15%. So none of the cells after the initial quality control was used to do the analysis, and this helped us to manage better the range of the real mitochondrial content within the samples.
Okay, we have two more questions. One is from Samuel. So what can we find more of your work, such as publication packages or projects? So you guys are, sorry, just want to add one more comment here. So if you want to talk more with our speakers, you can raise your hand. I can try to to unmute you, so you can. I think it's easy for you guys to communicate also.
Yeah,
sorry,
like
Daniel.
All the packages are published as open source packages and are available through the CRAN repository. So it's just install.packages and the name of the package. And all the supplementary information and the testing datasets are placed on GitHub. They are all open and available for anyone to test and play with the data. And yeah, I guess that will be the answer for that question.
Okay, one more question from Wanqing. Following up, this is basically follow-up question with with Ashish's question. Does the correction work well for data set generating using different single cell RNA-seq protocol like split-seq and smart-seq?
Yes, like it works very well. Like for example, in this project where we trace back the tumor cells from the patients to their more similar cell lines, well, the atlas of cancer cell lines was done using smart-seq approaches, and the cells from the patient tumors were sequenced using the tenet's chromium system. So what we did was that we first cross-validated the mapping function using different technologies to map the cells to the original atlas of cell lines. So we took MCF7 from a different lab and using a different technique to map them back into the atlas and see if the batch effect could be removed and the identification could be correct. We found that it is accurate with the 90% of precision.